Pretty thighs, shawty, she a dime. Demon girl, evil eyes, she be telling lies. Slim waist.
So today it is my pleasure to interview Dr. Martin Chalfie, the 2008 a Nobel Prize winner in chemistry. So Dr. Chalfie, thank you for being part of this episode. So as a spearheader in your field, can you bring us back and tell the people who are watching or just tuning in about your background?
My background, I was born in Chicago, 1947. Grew up in Chicago and then the suburbs of Chicago, specifically Skokie, Illinois. Went to high school in Skokie and left for college. I went to Harvard. I was an undergraduate there. Graduated not knowing what in the world I was going to do, and then spent several years doing odd jobs and doing a very odd job. I was a high school teacher for a couple of years. And while I was teaching high school, I had a chance over the summer to do some research that got me excited about doing research, also got me the confidence that I felt I could actually do work in a laboratory. And I applied and got into graduate school. From there, I went to do a postdoc at the Laboratory of Molecular Biology in Cambridge, England, in the group that was headed by Sidney Brenner, although Sidney wasn't involved that much in that. He just basically let people do what they wanted to do. And but it was a wonderful place to work, great people to talk with, any equipment or supplies you might ever need. And so it was up to you. And it was a very, very wonderful environment. From there, in 1982, I went to take on a faculty position here at Columbia, and I've been here ever since.
So I just want to ask more about your, I guess from your bio, you mentioned that you lost your fellowship when you went to Harvard. So my question is, how do you lose it, and what we have done? If you had a chance, like what would you have done something different if you could just redo the whole process?
When I was about to walk into my oral exam for my PhD, some of my fellow students, or people that had already gone through the exam, pulled me aside and said, "We have to warn you about a question that somebody's going to ask you. They're going to say to you, now that you've completed all the work for your PhD, how would you go and do it differently? And if you answered that question by saying, well, now I realized that I was actually addressing the wrong problem, I should have looked at this problem, or I should have done that," they were going to then tell you, okay, you fail. Now go and do the stuff that you should have done. So that was done so that I or anybody was going into their oral exam would be very frightened. How should I answer that question? After they let you stew for a couple of minutes, then they say, look, there's only one way to answer that question. The answer to how would you do it differently is I would have done it faster. So when you ask me, how would I have done it differently? There's no way of knowing what I would have done or not done. But to be honest about it, I don't think that I was really ready to do graduate work. I don't really think I had a lot of misconceptions about how to do science, how to interact with people. And one of the biggest misconceptions was that somehow it had to be something innate in me that I had to do on my own. And I think that was a very bad idea, that I was somehow, that real scientists came up with these ideas all on their own. They didn't ask for help, they didn't interact with people. And now I realize that that was exactly the wrong thing, is that the interactions are really important. The conversations with people are very important. And taking one's ego out of the whole equation is also very important, at least for me. How other people do this is absolutely up to them. And I don't know if any one way of addressing things is the right way of doing it. But for me, I think I didn't really know how to be involved in a project. And that was important. I was basically on my own. And frankly, I don't think I had the confidence that I think one needs that to keep going forward. I was easily discouraged.
Mhm.
And that's a problem. You get discouraged. My way of reacting to being discouraged, I had a summer experience working in the lab after my junior year in college where nothing worked. And my response to that was, okay, I quit. I'm going to do something else. And so I didn't overcome adversity. I just gave into it. And I was just fortunate that several years later, I had another experience in the lab. I was able to come up with an idea. I was able to find people to help me see if that idea was true or not. And it led to my first published paper. But the difference there was I was talking to people. I was contributing, but I wasn't doing it alone. And I think that was the important thing that I learned.
I see. So, I mean, I want to go back to your point about that you worked at some odd jobs. So, I think at every job, people can learn at least something, even if it's an odd job or it's research. So, what do you learn during the time when you're working at some odd jobs?
Well, by odd jobs, I mean very odd jobs.
Yeah.
I did a whole series of jobs for about 1 month each. I was a janitor for a month. I was hired by a company that was doing research on setting up a junior college curriculum for people to learn how to repair biomedical equipment. So I would interview people in hospitals. What equipment do you need fixed? What are the requirements you need of somebody to do that? That took 1 month. That was all I was hired for. It was one unfortunate thing that happened that year was my father had colon cancer, had to have an operation, and my mother and father and my grandmother worked in a family business. My grandmother had started in 1933. It was a dress manufacturing company. And my mother ran the office, the factory where they made dresses. My father and an uncle were the salespeople. And so twice a year, my father would go out and drive along a approximately 5,000-mile route selling dresses. And he got ill, and I had finished college, and I didn't have another one of these 1 month jobs. So that's what I did for 1 month. I went off and I was a dress salesman. I would go to stores that, and I did learn a lot from that, actually. I learned a couple of wonderful things. First, I learned, I think probably the most important thing was I learned about my father's feelings about how one sells. And my father had a very interesting idea. His idea was that he would sell to only one store in every city. There wouldn't be competition. And he did not want to sell anyone, put something over on anyone. He wanted to be able to go back year after year after year and have these people as his customers. And that's exactly what happened. And so I could do it because I didn't know anything about selling dresses, but I could drive a car and I could drive up to the store and I would bring the samples in, and the owner of the store, these were all usually very small stores, the owner of the store, the salespeople from the store would look at the dresses and they'd say, you know, Mrs. Brown is really going to like this thing in red, and Mrs. Jones is going to like that one in green, and so on. And they would pick out their continuing customers on how they would, of who they thought this would be good. Then they buy those things for the people they knew they were going to sell to, and then they would show them. And other people would walk into their stores and say, "Oh, you know, I like that. Can you get that in purple? Can you get that in?" So, and these were not cheap clothes. These were clothes that were made for that time. This was 1970. Businesswomen, wives of businessmen, professional people, and they would, and so there was no selling involved. They just pointed to the things they want, and I wrote down what they had, and I went from one store to the next. Every once in a while, someone would say, "What did other people buy?" And I was able to tell them. But I was able to do it only because my father had built up the rapport with these people and had their appreciation, and they knew exactly what they were going to be getting. There was so the idea of selling something, putting something over, that wasn't part of it, and it gave me went about the work that he did.
I see. So, like,
and then I taught high school.
Okay. So, let's talk about your when you taught high school. So I know you were a high school teacher, then you became a researcher. So, what's the biggest difference between being a teacher and a scientist? Because I know scientists also teach as well, you know.
So I think teaching high school did give me experience talking with people, preparing lessons, making sure that I could explain what I was doing, try to understand that, try to get over what my understanding of an idea was. I found that to be very rewarding. I didn't really like all the administrative hassles. Once when it was in a faculty meeting and there was a discussion about some idea or some procedure that we were going to do, and I volunteered an idea that seemed to me to be perfectly logical, and had the head of the school look at me and say, "Marty, that sounds right. We're never going to do that." And so being ignored with what I thought was a good idea, that was not very pleasant. But the actual teaching itself was fine, and I'm a little bit of a ham, so I sort of enjoyed it. I think that's good. I did notice one interesting difference between doing research, which, as I said, I did in the summer between the 2 years that I taught high school, and the teaching. When I was teaching, I and the other faculty members, whenever we had some free time, we would go into the faculty room and have a break. And our break usually amounted to reading the newspaper or magazine or grading exams or papers, but we didn't really talk to each other that much. In the lab, however, where I was working on my own or doing the experiments, it was often a real joy when someone else came in and we would start talking. So I think people have this idea about scientists as not being gregarious or being loners. And I found it exactly the opposite. Whenever someone walked into my room in the lab, I found that we were doing a lot of talking. But the faculty room, when I was teaching, people weren't talking. And I think it was because they had just been spending hours talking to students. So I've always found that a little bit ironic that I had more conversations when I was working in the lab with people than I did with my fellow faculty members at the high school. I should say I got friends in both places. So it wasn't that some people were unfriendly or not. I think people that were teaching who had been talking the whole time just wanted some quiet.
So let me I just want to ask, I know you graduated college, but I know like now teachers have to get like a diploma to teach or something. Did you get one?
I'm a teaching diploma, is that what you just said?
Well, it's like this because I know I was in high school. Like my teachers, they have to get their bachelor's. They have to go to some university to get like a teaching diploma that says like, oh, you're able to teach.
Yeah, no, I was incompetent.
Oh,
I did not have that. I actually taught at a private school that didn't have those requirements. Had I wanted to teach in a public school, I would have had to have some sort of teaching certificate. And I toyed with the idea for a while, but
okay.
then decided not to do that. So no, I did not have any sort of teaching credentials.
Mm-hmm.
You're
So
going to have to talk to my students now to see if I still need them.
so why do you move from Boston to the UK? Because I mean Boston is like the biotech hub and for research. So why do you move to the UK?
So this is a wonderful general question. So first of all, it wasn't a biotech hub when I was a graduate student. That was still in the future. Although it was a wonderful place for research. There were great universities, as there still are. And it was a wonderful place. But this is a general question I think graduate students often address, and that is, okay, I've just finished my PhD. Should I continue doing the same thing that I've done in the past, or should I go off and do something new and different? And I'm very much of the school that says, go off and do something different. In part, it goes back to this idea of having confidence in oneself. By the time one finishes a PhD, I feel that they have become an expert in that area. And that's very comfortable, being someone that knows the literature, knows the ideas in the field, knows the approaches that people are making. But maybe that's too comfortable. That it's really nice to know maybe you can do something else. Maybe you can address a problem in a different way. And so going somewhere else to interact with a whole bunch of new people, getting new approaches and new thoughts about how they're doing things, I think can be a very useful thing. And then, to tell you the truth, it was England. I was going to be able to go and do my postdoc in England. It seemed like a wonderful idea to be able to get away and go somewhere completely different.
I see.
And in retrospect, the LMB, the Laboratory of Molecular Biology in Cambridge, England, is one of the very, very finest research organizations in the world. And so it was a fantastic opportunity. I'm not even sure I realized that when I first applied, but it was clearly going to be something new and different. And as it turned out, it was a spectacular experience.
So I meant from moving from the US to UK, do you see any lab culture like
It's
differences
hard
or
it's hard to say this in terms of a country.
Hmm.
That's that's going too far. Was there a difference in the laboratory of molecular biology with what I had experienced previously? The answer is yes. And I'd say the main thing was a real enthusiasm generally for science and excitement about being involved in the research. There's an example I like to give. One day at coffee, which was at 10:30 in the morning, time when people took a short break, someone came in and announced to people that they had just heard that the University of Cambridge University astronomy department had gotten the videos, the films of the Voyager 2 flyby of Jupiter, and they were going to show them in the astronomy department. And something like 35 or so molecular biologists get up and go to their cars and drive down to the astronomy department just to be able to see these movies because it seemed exciting. If a book came out, there were a number of books by Stephen Jay Gould. One was Phylogeny and Autogeny. Maybe I have the title backwards. Almost everyone seemed to read them. If there was a new general science book, people were interested in science. There was a general excitement all the time about what people were doing, and people talk science all the time. And that was different from what I experienced in other places where people were just as committed. But this was special. This was very different. So I'm not even sure that many other places in England would have been the same way as well. But for me, this was fantastic. And it may also have, in large part, been because of the way Sidney Brenner, who I ostensibly did my postdoc with, organized his lab. He didn't really organize his lab. He just let people come. And there were all these wonderful people to interact with. And we were able to do it. It was really sort of a scientific heaven. It was quite wonderful.
yes, and I want to go back a bit. I know you were on the swim team at Harvard, so like what event were you doing and like what was your fastest time or
Oh, that's a good question. I was a butterflyer.
oh
I
okay.
did some freestyle. I couldn't swim breaststroke to save my life, so I was never did the individual medley except to humorous effect. But no, I was a butterflyer. I can't tell you what my fastest time was. I think for the 200-yard butterfly, it was something like 2 minutes one plus seconds. I can't. It's been a long time. I I don't know. The swimming team was an important part of my life, and I can spout off a little bit about that. I feel that for many people, I've seen this here at Columbia. I've seen this. My daughter has now gone through college. I think almost everybody I've ever seen going through college, and certainly myself, college is a shock. It's a shock in the sense that we take people and usually ship them off somewhere and say, forget if you had brothers and sisters, now you have all new siblings, although we call them roommates. And your friends, yes, you can try to keep up with your friends by social media, but frankly, you're going to have to make all new friends. And there are all sorts of differences in the way you're going to do your academics. You probably in high school went and were in the same classes with many of your friends. You went from one class to another. That's highly unlikely ever to happen again. You're going to be doing it on your own. You also spent your whole day in your high school. That's not going to happen. You're going to have a lot more time to yourself, which you're going to have to figure out how to deal with. And you're 18. We now consider you an adult. And all those adult things that now that people have to navigate, sex, drugs, alcohol, all sorts of things, are things that now you're going to have to navigate in earnest. And then after we do all of that to people, we say to them, oh, how are your grades? You know, we ask an awful lot. And I think that for many people, college is a time where if they're not fortunate, they can feel quite isolated. How can one counter that in all of this? One is to have something that you're not being evaluated on, but you enjoy and that you interact with, and you have I would put it, a home. For me, the swimming team was the home. It was a place where I didn't care what my grades were. I was just part of the group. It was something I did. It was companionship. People join a cappella groups. They join theater groups. They are in chess clubs. They are working in a lab. Can be a wonderful way of grounding oneself and having meaningful interactions at the same time as you're going to college and dealing with all the other things of growing up and everything else that you have to do. So for me, being on the swimming team was a very important part of my coping with what I think for almost everyone is a real shock of going to a new place, interacting with people in a different way, and starting to navigate being an adult. So that's why it was an important thing for me. My times were not that good, and I was not that great of an athlete, but it was enjoyable.
So, no, what got you interested in C. elegans?
So when I was doing my graduate work, I had a roommate who worked as a technician for the first American lab working on C. elegans. And this was a lab by Sam Ward. And so I got a little bit of a taste of that, hearing about it. But I really got interested in working with C. elegans because a high school friend of mine who was doing that, working in Sidney Brenner's lab on C. elegans, came by visiting, and we were talking about some science that he was interested in. And he's a professor at MIT, Bob Horvitz. Bob shared the medicine physiology Nobel Prize in 2002 based on the work, at least in part, that he and did in when he was a postdoc. And so we talked about the postdoc that he was doing. And it was a time when I was being a little bit, I had a wonderful experience in my graduate work. I had worked with a spectacular person, Bob Perlman, who we were working on the release and the synthesis of noradrenaline, norepinephrine, catecholamines in general. And I had done a lot of work. He had been a wonderful person to work with. We still are good friends. And I went looking in that area, and I felt to myself that I would just be doing more of the same thing, that he had taught me so well, that I had learned so many things. And maybe I didn't have the imagination to think of what new things I wanted to do, but I just felt there was something missing. And then when Bob Horvitz came by and was telling me about the work he was doing, I thought, this sounds interesting and exciting and different, and I'd be able to go to England. So I used that as a way of doing it. That was an exceptionally lucky thing to have happen, to be able to go and work at that time on C. elegans, because there were not many people. In fact, just before leaving for my postdoc, I participated in what we now call the first international worm meeting. I think there were about 125 or so people, and that included technicians and undergraduates that were just starting to work on this animal. And it was quite wonderful to be involved in something new and different. So I feel I lucked out. And then I really was fortunate on the way to that meeting, which was the marine biology lab at Woods Hole. I had picked up Bob Horvitz at the airport, and we were driving to Woods Hole, and he said to me, "Marty, you should take a look at the work that John Sulston is going to be talking about, because John's not going to follow up on it. And you might consider that as a very interesting project." What John Sulston had found were the very first mutants that were insensitive to touch. And it was a wonderful opportunity to work on something I had never thought of before, and that is sensory biology. How do we detect the world around us? What are the mechanisms of that? And in particular, mechanical sensation. Mechanical sensation is something that really wasn't studied at all. And so this was an opportunity to use genetics in a really novel way. And so it was a wonderful opportunity. And I had this great conversation with John and decided if he wasn't going to do any more on this, that it would be wonderful for me to take up this and see where it went. And it went in many directions. And I was quite happy with that. So I was very fortunate to be able to do that.
Yeah, so my next question is about GFPs. So so how do you persuade a wife to cite her unpublished research? I mean, it just seems impossible, right? It's like no one's gonna say, all right, you know, it has to be, you have to pay her or do something. So
no, so people will understand what the background is in this. When we were about to publish our paper showing that GFP could be a very useful biological marker, I had already been sending samples to people, and I wanted to cite their successful unpublished work using GFP. to show that it was something more than just what we were doing in the lab. Because our paper that was published in 1994 in Science, that paper was not really the first time the world heard about our results with GFP. There is a newsletter that we have for the C. elegans field called the Worm Breeders Gazette. And for the October 1st, 1993 issue, I had written up a one-page summary saying, this is what it is. Here's some ideas that I have. If people want it, please get in touch. And so people were starting to get in touch, and we were sending out samples, and people were saying that they had successfully used GFP. With this in mind, I wanted to put those successful results in my paper. And as I asked people, can I cite your unpublished work? They said, of course, you gave us your stuff before you published. Of course you can. And my wife and I often joke around, and one of the things is that she wrote this wonderful letter that I included in my Nobel speech, in which she said, yes, you can cite our unpublished work. Now, parenthetically, GFP is useful because you can use it to look at where genes are expressed. That is, thinking very simply about a gene, it has two parts. It has a coding part that says what the product should be, whether an RNA or ultimately a protein, and then a regulatory part that says where, when, and how much it should be made. And we were using GFP as a reporter, so replacing the coding sequence with GFP. And so that would be called a transcriptional reporter. But another thing that could be done is to add GFP not with just the regulatory part of the gene, but with the coding part as well, so that you make a fusion protein that has the original protein now with GFP added to it. And my wife was the first person to do that experiment, to make a protein or translational fusion. And so when I asked her, her name is Tula Hazelrig, when I asked Tula, can I use your result? She said, as the others had, of course, and wrote a very simple one-line permission to Science magazine, so they had it on their record. However, to me, she sent a completely different letter. And in the letter, she said, in order for you to do this, here's what you need to do. You have to take, you have to make coffee every Saturday for 2 months. You have to prepare a special French dinner, and you have to take out the garbage nightly for a month. And I thought this was hilarious. This was not the letter that was sent to Science. But I love showing, for a while there, I used to show it in my talks because it's amusing. Mm-hmm. She's a very amusing person.
So, Ahmed, how do you get, what event, made you want to start working with GFPs?
So that's an interesting question in the following sense. How do you get any ideas? I think for myself, it's because of what I was at the right place at the right time. Mm. I went to so what do I mean by the right time? By the right time, I have to tell you a little bit about what we were doing. So what we were doing at that time is we were still working, as we still are, on the sense of touch. We had done extensive mutageneses. We had identified a series of genes that could be mutated to cause touch insensitivity. And we were beginning in the middle 1980s, or sort of at the end of the 1980s, we were starting to clone those genes to find out what the products were. This was an exceptionally exciting time to be able not just to have the genetics, but to be able to actually get at the gene. And when one cloned a gene, one of the first questions you wanted to ask was, where is that gene active? Is it active in the cells that you think you're studying, or is it maybe active somewhere else? And that might give you some insight into how it's working. And so we wanted to know the answer to the question, where is a particular gene turned on? And there were many ways of answering that question. DNA makes RNA makes protein, so you could do in situ hybridization to the RNA. You could make an antibody to the protein and see where the antibody stained the animal. Look at where the protein was. Or you could use a reporter, either a translational or a transcriptional reporter. And one of the commonly used ones was the product of the LAC-Z gene from E. coli, beta-galactosidase. There was a wonderful substrate called XGAL that when the enzyme was present, it would produce this beautiful blue precipitate. And so you could see which cells made the beta-galactosidase. Now, all of these methods, in situ hybridization, antibodies, using beta-galactosidase, all of these required fixing the preparation and permeabilizing it to get the material in. But they gave the answer. So it's not a story of, oh, we didn't know how to get the answer. There were lots of ways of getting the answer. But these are the experiments we were doing. So the right time, part of the idea of the right time was, I am asking questions about where in an animal the particular genes are turned on. The second part is the animal that we use, which was C. elegans. C. elegans, as I would tell audiences whenever I would talk about the animal, is transparent. So those are the two ideas. Where are genes expressed? And I work on a transparent organism. In 1989, I went to a lunchtime seminar here in my department, and the speaker, Paul Brehm, started talking about the jellyfish, Aquaria victoria, and similar organisms that I knew made this wonderful bioluminescent protein called aquarin, which was activated by calcium. That was something that I had heard of. But what I had not heard of was the fact that in addition, the jellyfish and some of these other organisms had another protein, the green fluorescent protein. This was the first time I heard about GFP. And going back to what I just said, I work on a transparent animal, and I'm using reporters to see where genes are active. And I realized at that point that, one, this would be a lot of fun, and two, we'd be able to look in living animals. We wouldn't have to permeabilize it because all we would have to do is shine blue light on it and see wherever the GFP was by seeing where it was green. So it was the combination of the type of experiment we were doing and the fact that we were doing it in an organism that was transparent. This is the way I think, and that we were doing some molecular biology. I think that most of the other people in that seminar were people that had not yet started doing molecular biology. It was mainly people interested in neuroscience. They were using cells in culture, but they weren't looking at gene expression. And they were also looking at organisms that were not transparent.
Mm.
But I got exceptionally excited about that and ignored the rest of the seminar, just thinking about what would happen if all of this actually worked out. And fortunately, it did.
See. So, my next question is, GFPs, I know you're part of the development. Are we seeing it used in our drugs or vaccines?
So, there are the use of fluorescent molecules is quite extensive. I mean, people use it a lot. It is not itself a drug. I have heard of people that, as they have tested, every once in a while you read about attempts of being able to get viruses or bacteria to home in on cancer cells, for example. Well, how do you know that that happens? How do you know that that actually will take place? So in some early stage clinical trials, people will take the virus and put GFP in it, or the gene for GFP, so it'll be expressed where that virus attacks a cell. And so one can see, did it attack the cancer cells? Is that a way of labeling the cancer cells? So it's not that it's unheard of, but as far as I know, it is not itself. It can be used as a tool, as a way of studying the process in the course of understanding how things are distributed, how they work, and so on. But it's not itself that I know of a tool that is used as for some property that it has. Now, there may very well be ways of altering things. I know that Roger Chen, when he was alive, was very interested in the idea of being able to get a fluorescent marker to localize that to cancer cells. And he had a very good point, and that was, when a surgeon goes in and she wants to remove a cancer, she removes what she can see. And if it's just has to be a large tumor or a noticeable tumor, then that's what's going to be taken out. And more than that, they will take out the area around that to just make sure they get everything. But what if you could make it fluorescent? If you could make the cancer fluorescent, then using that, you could look for even the small patches of fluorescent cells. But I'm not sure for that you need GFP to do that. You need a fluorescent molecule. You need a molecule to do it. But you don't need GFP to do that.
So I know you won the 2008 Nobel Prize. So I asked three of the previous people, the winners who came on the show. So how do you find out you actually won this prize?
For me, it was I slept through it.
Oh.
So I did not. The story I like to say is that the night before. The thing is, if I think people are honest about this, one of the very nice things, and one of maybe also a little horrible things, is sometimes you will have friends that will come to you and say, we liked your work so much, we think it might be honored by the Nobel Prize. Now, that makes your head swell a little bit that that's going to maybe happen. And if you're an idiot, and I put myself firmly in that category, you might lose some sleep waiting for that phone call.
Oh.
That does not come. So then in 2008, the night before the chemistry prize was announced, I actually got an email from a student. And the student said that he wanted to have a copy, a PDF copy of my science paper on GFP. And in his email, he said, he and his friends had been talking, and they wondered about who was going to get the chemistry prize the next day. And in his letter, he said, and we think that the prize tomorrow should go to Shimamura and Chen. And I looked to see if he included my name in that list, and he didn't. So it was just these other two people. So I wrote him, and I said, you can add a third person, and I named some people that could be added, because in fact it's many more than three people that have made GFP important. And I sent them the PDF, and I showed this email to my wife, who, as I've told you, did the second really important experiment by making the fusion protein with GFP. And frankly, I think she should have been honored as well. And I showed her this, and she remembered that time that I had stayed up late and lost sleep, being an idiot. And so she said, Marty, I just have to tell you, many people use GFP. It's been a very useful tool. It's a wonderful reagent and everything. But I got to tell you the truth. It is never going to get a Nobel Prize. So just go to sleep, which is what I did. So I went to sleep. At 10 after 6 in the morning, I woke up. We live in an apartment building. I heard what I thought was the upstairs neighbor's phone ringing, and I was getting a little upset that he didn't answer his phone. Our phone was between the bedroom and where the phone was were two closed doors, so I didn't think it was our phone. And I woke up, and it was 10 after 6, and I realized, they must have announced who got the chemistry Nobel Prize. I wonder who it is. And so I had my laptop next to my our bed, and I opened up the laptop and I looked at it, and there was my name. And that was very surprising. And I turned to my wife and I said, I woke her up. I said, it happened. And she thought I meant that our teenage daughter had overslept going to high school. And so she jumped up to help our daughter. And I said, no, not that. And I showed her the laptop. And we were pretty happy about that. So that's how I actually found out about it. I found out about it by going online and seeing my name listed as one of the recipients.
I see. So I know there are two other people who won the award. So have you met them, collaborated with them?
So the two other people that I shared the prize with were Osamu Shimamura and Roger Chen. Now, Osamu was the person who, working on bioluminescence, discovered green fluorescent protein. This is a very important and really underappreciated discovery, that people knew that there was bioluminescence, that there were molecules, enzymes that could generate light. But I don't think anybody had an idea that there could be biologically fluorescent molecules. And that's what he discovered, in his case, GFP. And the reason was he had discovered aquarin, the bioluminescent molecule, and realized that it produced blue light and not the green light that the jellyfish had. And so he reasoned there must be another protein that actually does that conversion. And then he did a lot of biochemistry on that molecule. So Osamu Shimamura shared the prize because of his discovery of the molecule. Without that, nothing would have happened. I was honored because I said, this is a wonderful tool, and here's an example of using it. we can use this as a biological marker. So I introduced the idea of GFP, fluorescent proteins, as biological markers. Roger, who had previously done a lot of wonderful chemistry on fluorescent molecules and applying them to various biological problems, did a lot of wonderful experiments modifying GFP, changing its excitation spectrum, changing its emission spectra, being able to make what are called FRET or Forster or fluorescence resonance energy transfer pairs, to be able to use them as biological monitors of various aspects like calcium in cells or redox potential or pH and other aspects. And he did a lot of modifications of GFP and subsequent other fluorescent molecules. And so it's the discovery, the introduction of the use, and then the modification of the work.
Okay. So I've always wanted to ask this, during the ceremony, do they give you like the real medal, or is it some type of fake one or replica?
So on December 10th, which is the anniversary of Nobel's death, they have the ceremony. Starts at 4 p.m, as I remember it. It's at the concert hall. On the stage are the laureates, the royal family from Sweden, and the electors. And they announce each one of the winners in turn. And as your name is, short paragraph is read about you, you go up and you accept the proclamation and the medal from the King of Sweden. And you then shake hands with the king, you bow to the king, bow to the electors, and turn to the audience and bow to the audience, and then go back to your seat. And the next person does that. At the end of the ceremony, they take it away from you.
Oh.
And the reason they take it away is that the proclamation and medal are then displayed for people to see. And you don't get it back until 2 days later on the 12th of December. And that's when you get it. But you are handed the real medal at the time. And it's a medal in a box. There is no ribbon around it or anything like that. It's just the medal, which is quite nice.
Let's see. Okay, so I know in 2015 you signed a climate change agreement for the final day of the 65th Lindo meeting. So I've asked other scientists, like, do you think there should be like a climate change project, kind of like the Human General Project, where like every scientist works together and try to solve climate change?
Well, I think it is an exceptionally important problem. I think that the answer is I'm not sure every single person must do that. I'm very against saying everybody has to do it, but this is a major problem. And I think as more people work on it, we get to see what the dimensions of this problem are. I'll tell you my latest concern about all of this, which I think is an important concern. And I'm going to give you an analogy first. The analogy is, understandably, there are patient advocacy groups that want to see cures for diseases. I understand that. I support what they do. They often do spectacularly good work and support wonderful research. But I think it's important to remind people that the breakthroughs in understanding disease often do not come from studying the disease. They come from understanding the foundational biology underlying organismal development and function. And to say we are only going to study, pick your favorite disease, by studying people with that disease is losing an opportunity. And that's an opportunity to get new perspectives, to have new approaches. Look at what we have just gone through in terms of the pandemic with COVID. The messenger RNA vaccine, the mRNA vaccines, are a completely new, different way of approaching vaccinating people in a way that saves millions of lives. But that required understanding of an enormous amount, decades-long work on how messenger RNAs are made, how they're stabilized, how all sorts of aspects of their biology. And without that basic understanding, the vaccines could not have been made as rapidly as they were, and saving so many people. So that's my analogy. In talking with a friend of mine, Manu Prakash, who's at Stanford, who's a wonderful scientist, and he's made me realize that climate is the same issue. We often, if you listen to the news, you hear people talk, they often talk about climate as something we have to do something about. It's an engineering problem. It's a problem of mitigation or carbon sequestration or management of the land or something, or getting rid of cars, all of which is good. But to me, that's now equivalent to trying to treat the disease by only looking at the disease. Right. We need to know more of the fundamental science that underlies how our planet copes with all of these changes. The carbon cycle in the ocean, how the atmosphere takes adjusted to these things. We need to have more fundamental research to underlie our understanding of the issues that are involved in climate change. And at least talking with Manu, there are many, many questions that we have never asked. So I think, so I'm going to expand what you're saying. I think people should be working on all aspects of this, just like for a disease, you work on all aspects of a disease. I think people should be working on all aspects. Not all people, but people should be working on all aspects of things relating to climate. And some of those are going to be the basic science that we don't even realize now are going to be important for understanding and maybe mitigating this real problem that we have with climate change right now. So that's my answer.
All right. Is there any advice that you received during your career that shaped your success?
Not really. I think the basic, the advice that I would give myself is it's a science. It's the enthusiasm that you show for the science. It's your involvement that's important. And I think that is probably the most important aspect. Doing science is not a zero-sum game. There's not somebody at the top and somebody else. Anyone can do they can make a contribution. And if you want to see an example of that, go to any science fair, any of the high school science fairs or so, and look at what students are doing right now. It's astonishing. There are so many problems out there. It's being involved in doing this. I'm going to say I'm going to give you an example. For every so often, I go to the ISEF, the International Science and Engineering Fair, which is the culminating high school science fair for the whole world. They usually get 1,700, now more students from a lot of different countries, from every state in the United States. And it's just really wonderful. And one day I was walking among the exhibits, and I stopped by one. I don't know why I stopped. There was a young woman there, a senior in high school. And I said, "Tell me about your project." And she started the discussion. She started telling me about her project in a way I don't think anyone would have expected or anyone has ever heard of in this example. Her first sentence is, "I'm a ballerina." Not what you expect for a science fair project. Okay. And she says, and the problem that people working at very high levels, which she apparently was in ballet, is that the shoes actually last only one performance. They have a cardboard sole that gives them the flexibility that they need, but through one performance, one dance performance, they become impossible to use anymore. And so that means that people at that high level are shelling out hundreds of dollars buying shoes all the time so they can perform. And so she said, so my project was to come up with a better sole for ballet shoes. And she said, I researched the materials that are available, and I found this carbon composite, I think she said, and I realized that if I used 3-ply of that, it was exactly the perfect flexibility. And this time it lasts for months. And that was her project. And to me, this was wonderful because she had seen a problem. She had researched it and thought about it, did experiments to determine what was the best thing, and came up with a solution. And so for me, that enthusiasm, that being aware of the problems that are all around us, I think those are fantastic things. I was very, very impressed. I would have taken her in my lab instantly. But it was really wonderful to see someone be excited about a problem that they had come up with. That doesn't always have to happen, but it was nice to see. So to me, I think the advice is be involved, be committed to doing it, be enthusiastic about it. It's a lot more fun being that way anyway.
So, I've asked a lot of questions. Is there anything else that you want to touch upon that we never talked about in this?
No, I think this is about it. I probably talked too much as it is.
So thank you, Dr. Chalfie, for being part of this episode and giving me the opportunity to talk to you so people can know more about GFPs.
Terrific. Take care.